Good morning children, I am Isis. Now I am going to teach you the regulation of gene expression. Normally, the amount and time of formation of gene products can be controlled according to the requirements of cell. And this process of controlling the time and amount of formation of gene products according to the requirement of cell is known as regulation of gene expression. In case of eukaryotes, there are multiple levels of regulation of gene expression we can see. That means in different levels, the regulation or the amount of or the time of formation of gene can be controlled at different levels in eukaryotes. We can see which are the levels on which the regulation of gene expression can be done in eukaryotes. So the first level is transcription level. Actually, this happens in eukaryotes. Transcription level. So, at the time of transcription or during the formation of hRNA, there is a chance of controlling of gene products or the formation of gene products. The amount and time can be controlled at the level of transcription in case of eukaryotes. And the second level is during processing. During processing. As you know, there is a post-transcriptional modifications called splicing. Splicing that include capping and tailing. At the time of processing also, there is a chance of regulation of gene expression in case of eukaryotes. And the third level is at the time of transportation of mRNA from nucleus to cytoplasm. So, transportation level. So, at the time of transportation of mRNA from nucleus to cytoplasm also there is a chance of controlling the amount and time of gene products. And at last, at translation level also, there is a regulation of gene expression. It's all applicable. These are all applicable in case of eukaryotes. So we can say there is a multiple level of regulation of gene expression in case of eukaryotes. Is it clear about the regulation of gene expression? In case of eukaryotes, there is a multiple levels of regulation of gene expression. But in case of prokaryotes, this regulation of gene expression is heavily influenced by environmental factors or availability of other resources, etc. Mainly, the regulation of gene expression is done by a protein called a repressor. So, in case of prokaryotes, the regulation of gene expression is done by a protein called a repressor. That I will teach you. So, here in prokaryotes, as I told you, there is many cistrons are there in the structural gene. That's why it is called a polycistronic. So, there is a mechanism in the regulation of gene expression in case of prokaryotes, and that mechanism is called operon. So, operon. Operon is a group of structural gene that can be regulated by other sites of gene, or we can say that can be regulated by a regulatory mechanism. Okay, that is called operon. So, operon consists of, or operon is a group of structural genes, and that is regulated by a regulatory mechanism. or we can say a group of structural genes are there as it is polycystic and it is regulated by or controlled by other sites of genes or regulatory mechanisms. So the structural genes and these regulatory mechanisms are collectively called operon. Okay. And now we can see what are the types of operon. So here, types of operon. There are two types of operon. They are inducible operon and next one is suppressible operon. So inducible operon is always or normally it is in off mode and it can be turned on mode. Okay. So normally it is in off mode and can be turned on. Okay. But here suppressible operon is normally in on mode. Normally in on mode and can be turned as off mode. Okay. So here in inducible operon, example is Lac operon. Lac operon is an example for inducible operon, and here suppressible operon, tryptophan operon. Tryptophan operon. Here we have to study this Lac operon, very important area. Lac operon. Lac operon is an inducible operon found in prokaryotes. We can see the Lac operon in detail. Now we can see the Lac operon. Actually, the operon concept or the concept of operon was proposed by the scientists Jacob and Monod. Jacob and Monod. And this Lac operon explains the metabolism of lactose in prokaryotes, especially in bacteria. And in bacterium, this Lac operon is used for lactose consumption. This Lac operon is a unit of prokaryotic gene expression. We can call it like that. It's a unit of prokaryotic gene expression. Prokaryotic gene expression. And it was proposed by Jacob and Monod. So we can define Lac operon as, or we can define operon as a unit of prokaryotic gene expression, which consists of a group of structural genes, a group of regulated structural genes and control elements, which are recognized by the regulatory gene products. So that is operon. So here the Lac operon is an inducible operon. Inducible operon means it normally remains as off mode and it can be turned as on mode. So I'll explain this Lac operon in two situations. So which are the two situations? Let's see. So before that, we have to see the components of Lac operon. So before getting into the details of Lac operon, how it remains switched off and turned switched on, before that we have to see the components of Lac operon. Components of Lac operon. Or we can say the components of operon. So for that, let's see. There is a part of the DNA segment, a gene is there called P gene or promoter gene. P stands for promoter gene. Okay, so this is the first segment, that is the P gene or promoter gene, and this is the place where the enzyme RNA polymerase can bind. So the P site is the place where the DNA dependent RNA polymerase enzyme can bind for transcription. Okay, and the next gene is I gene. I gene or regulatory gene or regulator gene. Regulator gene. So regulator gene is responsible for the production of a protein called a repressor. Okay, so it is produce a protein called a repressor. So this is I gene. And the third site is again P gene. So as I told you, P gene is the binding site of RNA polymerase enzyme. RNA polymerase enzyme. Here also the binding site of enzyme. RNA polymerase enzyme. Okay. And after that we can see another gene, that is operator gene. O gene. O gene is the operator gene. So these are the components of Lac operon. So the first is P gene, I gene, again P gene, and then O gene. O gene is a segment of this Lac operon or gene of this Lac operon which can control the expression of structural gene. So the O gene is followed by structural genes, and that structural genes are under the control of this operator gene. Is it clear? So operator gene is a part of the Lac operon or a gene of the Lac operon which can regulate the expression of structural genes. So next is, we can write regulate or control structural genes. Regulate or control structural genes. Next we can see the part of the structural gene. Actually here in prokaryotes there are many structural genes, I told you, that's why it is called the polycistronic. But in case of eukaryotes only one cistronic is there, that's why it is monocistronic. In the structural gene we can see many cistrons or many structural genes in prokaryotes there are. So here we can see three structural genes. They are Z gene, Y gene and A gene. So these three are called structural genes. Okay. Now we can see. Okay, the first one, Z gene is responsible for the production of an enzyme beta galactosidase. Beta galactosidase enzyme. So the Z, Y and A genes are called structural genes. Three structural genes are there as it is polycistronic. So Z gene is responsible for the production of an enzyme beta galactosidase, and Y gene is responsible for the production of an enzyme permease. And A gene is responsible for the production of an enzyme transacetylase. So these three are structural genes. Structural genes in the operon. Okay. So this is the structure or components of a Lac operon. Next we can see the two situations under which the system remains off and the second situation in which the system can turn on. So in first situation, that is when lactose is absent, or we can say in off mode. What is happen? In off mode means lactose is absent. When lactose is absent, how it remains? in off mode. Suppose this is a membrane. So we can see. So this is the outside medium. So inside the prokaryotic cell membrane. Okay, suppose this is the cell membrane of the prokaryote and this is outside the medium. Outside, no lactose actually. No lactose here. So the lactose cannot enter into the membrane. Suppose this is the membrane of the prokaryotic cell. This is the membrane. Okay, suppose this is the membrane. And inner to the cell, we can see the components. Which are the components? P site, I site, again P site, then O site or O gene, then Z gene, Y gene and A gene. Okay. Here, this is the RNA polymerase enzyme binding site. Enzyme binding site. This is a regulator gene or inhibitory gene or I gene. Okay. Now we can see this inhibitory gene or regulator gene first undergo transcription. So by transcription it forms an mRNA. So by transcription process it undergoes transcription and form mRNA. This is the mRNA formed by the I gene after transcription. Then again this mRNA undergo the process of translation. So after translation it produces a protein and this protein is called the repressor. So the repressor protein. And this repressor can bind the O gene. Okay. So the repressor can bind the O gene. I will show you the binding. So here the repressor can bind the O gene. Okay. So what is the problem after binding the repressor around the O gene? So here the transcription of this structural gene occur when the RNA polymerase enzyme slide over there. So when they move along this sequence, there is a transcription of this structural gene, then it undergo translation, then form the proteins. And it is the transcription and translation are controlled by the operator gene, as I told you. But the repressor has high affinity here that can bind the O gene or operator gene. So this enzyme RNA polymerase cannot slide over here. It prevents the operator gene or the structural gene from transcription. So the system is switched off due to the binding of the repressor. And the repressor actually is a protein produced by the regulator gene or I gene or inhibitory gene. So clear? And now we can see, so this is a situation when lactose is absent in the cell membrane or in the prokaryotes. So the system remains off. There is no transcription of the structural genes and no translation also. Okay, and now we can see the second situation. Second situation means how it turned switched off. So on mode. On mode means when lactose is present. When lactose is present, it can turn into on mode. So again we can see this is a cell membrane. So inside we can see the cell. So here once more we can draw that operon, PG, IG, PG, OG, then ZG, YG, then AG. Okay. Here the outside medium, lactose is present. Lactose is present. So this lactose can enter into the cell due to the enzyme permease. Due to the enzyme permease, this lactose can enter into the prokaryotic cell from the outside medium. And here the same process occurs. What will happen? So the enzyme binds the promoter site. Then there is a transcription of I gene. So I gene transcribed to form an mRNA. Okay, then mRNA translate to form a repressor protein. You can write as a repressor. Okay, or protein. So it binds the operator when it is in off mode. So this is the condition of off mode. So here we can see when the lactose is entered into the cell. So this permease allow the entry of this lactose into the cell. The enzyme permease allows the enzyme or the lactose into the cell. Entry of lactose is allowed by the permease enzyme. Okay, so when the lactose is here, this lactose can bind the protein. So it prevents the repressor from binding the OG or operator gene. So we can see OG. So this repressor gene is bound by the lactose. Here the lactose acts as an inducer. So what is that? Inducer. So inducer is a chemical or substrate that can bind the repressor protein and prevents the repressor from binding the operator gene. Actually, this inducer is also a component of Lac operon. Okay, so among the components of Lac operon, we can say inducer also there as the components of Lac operon. Here the inducer is lactose. So here lactose acts as an inducer to bind the repressor and prevent the repressor from binding the operator. So now the operator gene is free here. So there is no repressor. So the enzyme RNA polymerase can slide over here. Then there is a transcription of these enzyme or these structural genes. So they transcribe to form mRNA. Okay, and they undergo translation also. They undergo translation. Is it clear? So there is no repressor. So the operator gene can control the structural gene and the RNA polymerase enzyme can slide over along the sequence of structural genes, and the Z, Y and A gene can undergo transcription and translation. Then they produce an enzyme, beta-galactosidase enzyme by the Z gene and permease by the Y gene and trans-acetylase by the A gene. Okay. Here, this beta-galactosidase enzyme convert this lactose into glucose and galactose. Glucose and galactose in prokaryotes. So, these are essential. So, there is a conversion of lactose into glucose and galactose in prokaryotic cell with the help of beta-galactosidase enzyme, and that is essential for the proper growth and development of the prokaryotes. So this is a situation in which the system remains on and the lactose is present here. That's why the system turned on. Is it clear about the Lac operon? So this is a picture. So here P site you can see I P cycle again, and O is now free here. That's why they can undergo transcription and translation, then form the enzymes beta-galactosidase, permease and transacetylases. So here you can see the repressor is bound by the inducer. Which is the inducer here? Lactose is the inducer. Here the lactose is present, so the system is switched on. Is it clear about Lac operon and the regulation of gene expression in prokaryotes and eukaryotes? That's all for today. Thank you.